Welcome to Science with Dr. T. Today we'll be looking at how to use the amino acid codon chart to translate mRNA to protein. Let's go over a few key terms and concepts first. Messenger RNA, or mRNA, is synthesized from DNA during transcription. Bases in mRNA are grouped in threes to form codons. During translation, these codons are read by the ribosome to synthesize an amino acid chain. That amino acid chain then folds into a protein that goes on to function in the cell. So how do we know which codon codes for which amino acid? The amino acid codon chart tells us codons are made of three letters. The first letter tells you which row in the chart you should look in. The second letter tells you which box in that row to focus on. And the third letter tells you which row in the box you will find the codon you're looking for. I don't think anyone expects you to remember all of these, but you should definitely know the abbreviations for the amino acids that are listed. And it may also be good to remember the codon for methionine, since it is the start codon for translation, and there's only one codon for it. So it's pretty easy to remember. It may also be helpful to remember the stop codons. These are what tell the translation machinery that it has reached the end of the sequence and to stop translation. For me, it's easier to remember the DNA sequence. So it would be TGA, TAG, and TAA. Because remember, there are no U's in DNA, so those U's would be replaced with T's. It's easier for me because when I was an undergrad student, we had toga parties. So I've always remembered the stop codons as toga, tag, and ta. All right, now let's put these concepts into practice. Practice problem number 1. What amino acid sequence is encoded by the sequence shown below? 5' UAC, GGU, GAU, 3'. Okay, so our first codon is UAC. If we look at our codon chart, the U tells us to look in this first row, the A tells us to look in that third box in that row, and the C tells us to look on the second line in that box. So the UAC codes for TYR, which is tyrosine. The next codon is G, GU, and this codes for glycine. The last codon is GAU, which codes for aspartic acid. So now we have our amino acid sequence. And how would we approach this if it was a multiple choice question? I always start with eliminating any answer choice that you know can't be right. We know that first amino acid is TYR, so that means we can get rid of answer choices B and C. Our second amino acid is GLY, so we can cross out D, which leaves A as our correct answer. All right, let's practice with one more problem. So what amino acid sequence is encoded by the sequence shown below? 3' UCC, GGA, GAU, 5'. Okay, so if you notice, this sequence is a little bit different than the first problem. We are given the RNA sequence here in the 3' to 5' direction. But translation happens in the 5' to 3' direction. So that's how we have to read the sequence. With that, our first codon is UAG. If we look at our codon chart, the U tells us to look in this first row, the A tells us to look in that third box in that row, and then the G tells us to look in the last row of that box. So UAG codes for a stop codon. So remember, stop codons tell the translation machinery to stop translation. So if the first codon is a stop, then nothing else will come after it. So there are no amino acids made with this sequence. Right? This was a bit of a trick question. So you have to remember that the 5' and the 3' ends matter, okay? I hope this has been helpful. Make sure you practice on your own with a few different sequences until you get the hang of it. And thank you so much for tuning in. Remember to like our videos, subscribe to the channel, and share. Next time, we'll take a look at DNA mutations.